And we'll start by introducing Professor Jesper Andersen, who many of you will know, who from the University of Copenhagen, who's done a tremendous amount of work on molecular and genomic data in cholangiocarcinoma. Jesper.
Thank you very much. I'm honored to be here and be back in UK again. I was here in January for the ICCA UK in Nottingham as well. So in line of actually the previous question, I think one of the really key things is that we force industry to pay for genomic workup in the clinical trials. And then we also, in one way or another, through the very hard legal setups that are between countries and now also between UK and Europe, try to make that data available for people like me and many others to actually go deep into that data and correlate it with some of the more historical data we have and the more basic research parts of questions you can ask with data today. And I will go into some of that in the presentation, because when I looked at the lineup of speakers yesterday, also talking about, as just presented, the DNA and RNA, I was thinking, so the molecular data and implications of genomics for cholangiocarcinoma for the patients, that Juan Walla and Angela Marca would probably take care of with all the approvals we have had in the last 3-4 years, at least the last 4 approvals that are all precision-based or targeted-based therapies. So I was thinking maybe to be a little bit more provocative, both going historical, but also provocative in the second part of the presentation. So first, on the background, mutations matter. It's a success story by the CCF, Stacy, and also Helen at AMF to really push that concept for the patients so they do get their biopsies profiled. And now we have, as I said, 4 different approved drugs, 3 in the FGFR fusions and IDH for the patients. But later I would like to put into the question, so how successful are these drugs actually, and how many people are left behind, or how many patients are left behind? So what is next from the targeted therapies and then into precision chemotherapy? Chiara Branconi introduced it in the ENSCA session, that we have a very tight collaboration on trying to push chemotherapy that most patients anyway still see, even though it seems old-fashioned in the era of precision and targeted therapies or individualized therapies, as we heard about yesterday. Chemotherapy is something that most patients still see. And should we actually put in precision in that setting as well? Who should actually get chemotherapy and who should not? Who would actually not benefit at all from getting chemotherapy, and what should they then get instead? So going historical, for the patients that think that the development in cholangiocarcinoma is going way too slow, and of course it is. It's a dreadful diagnosis to get. It still is. It is actually only approximately 10 years ago that the first genomic profile, it was a transcriptomic. It was in the era of arrays. It was not sequencing, for those that still remember that part. So it was probes. You put on a sample and you get some light in a scanner that indicated how much this gene is expressed. We actually spent 5 years of my postdoc time to accumulate these 104 samples of fresh frozen tissues. There was nobody that really stored these samples. They got thrown out after the diagnosis was done or put into FFPE, and the technologies did not allow us to interrogate FFPE or archival samples at that time. It had to be fresh frozen. So technology moves very, very fast, probably also faster than we can implement things to the clinic and for the patients to get the benefit of it, but that is still that's still how things are. So Illumina as a technology came online during those 5 years and made it possible for actually us to interrogate the transcriptome differences of these patients, to set up something as simple as a good and a poor prognosis class. That's followed up by many others since then, but the reality is it basically shows the same thing. You have a good prognosis class. There are some immune-related changes there in cytokines and interleukins, and in the poor prognosis class, you have all the mutations, or many of the mutations. You have all the receptor-tyrosine kinases that was spent 10 years of trying to target those with the TKIs, and most of those clinical trials failed, as we will see in a little while. And now we're coming back to those trials again, hopefully with a little more precision in the patient selection. Anyway, the networks are still the same as it was 10 years ago. It's still the same networks we see today that are changed between a normal setting and cholangiocarcinoma. Now it moved really, really fast, suddenly, from nothing in 2010-12 to 5-7 years following that with the NGS age, so next generation sequencing age, where a lot of different studies came online, studies coming from all over the world, not just from US, not just from China, but also from Europe and so forth in line of this work. So researchers also decided to look into cholangiocarcinoma in various different ways. So the Tiger LC, looking into it as a continuum, comparing HCC and intrahepatic cholangiocarcinoma, the two primary liver cancer subtypes in the liver, and actually discussing on patient material the continuum that we have known from mouse studies for quite a long time. TCDA came online with the multi-omics approaches, where multiple different types of data were investigated. Also, pan-cancer studies linking where intrahepatic cholangiocarcinoma was a part of, and linking the different pathways that are changed in line of other cancer types as well. So, in the point of how can you actually repurpose drugs from other approved entities. Epigenomics came online, and we did a study on just a few different driver mutations, the IDH that now have the targeted therapy, KRAS where we are not able to target currently, but maybe in the near future we might be able to, P53 that nobody can target so far, and then what we call the undetermined, and there we actually were a subset of those patients that were the FGFR2 fusions, so also are now 3 different approved targeted therapies. I'm not going to go into this study. I've talked about this countless times before, but the whole point is that for each of these subgroups of patients, they have a unique perturbation mechanisms, and that goes again in the different types of data that we have been looking at, if it's the transcriptome, the copy numbers, the epigenome, or the mutational base of across the genome. We see the same thing for these patients, so we are able to develop network vulnerabilities for them, and many of those actually are targetable if we repurpose drugs. Now I wanted to take this large meta-analysis that went a step further, also integrating histopathology into the meta-analysis of integrated genomics, to highlight also the differences in the epidemiology of this, or the ethnicity of these cases. So there, cluster 1, that includes the KRAS mutations and are mostly the large bile duct cases. We have Asian patients, whereas the small bile ducts or the peripheral bile duct patients are more cryptogenic and have the IDH, different epigenomic alterations, the FGFR2 fusions located, and they're mostly the European or the Caucasian descent. So there's huge differences in who we are talking about among the patients, and therefore that has to be taken into account when we do clinical trials as well. Now you will also see that we have the 5th success in cholangiocarcinoma that are here highlighted for more the Asian population, where you can where you have a high checkpoint PD-1, PD-L1, and that would indicate a potential for chemotherapy and durvalumab. You have the IDH for the more Caucasian group of patients for IDH and IDH mutated cases, so with TIP2, and then you have FGFR alterations and FGFR2 fusions with pemigatinib and the other approved therapies in that area. Now, the explosion that you saw on Chiara's slide around 2010 in publications also coincided with ABCO2 by Juan Wilder and John Bridgewater, the large clinical trial with 430 patients that indicated the use of chemotherapy in cholangiocarcinoma did not necessarily meet all the endpoints, and therefore it never really got approved, but it got adapted to all clinical, I can say, to all clinical departments worldwide, and are still the benchmark for therapy for most patients today. And that was back in 2010. Then it took a decade before we got to the successes that we have been talking about the last couple of days, and also talked about in a lot of other meetings. Now the question is, are we going to have a period of time again before we get to the next, and what is the next? So in that relation, I want to go to the more provocative part of my presentation. So why do patients not respond necessarily always to these targeted therapies when they are targeted? And what about the patients that do not have a target? So have we focused too much on mutations matters, letting driver mutations guide us, and completely forgotten that there are other things that are ongoing in these tumors and their development over time, and also in the reason for the background, such as the pro-inflammatory signals and the microenvironmental clues? We did not early on actually have the technologies to really detailed on large data go in to investigate these things, but that we have now, and that's what I want to highlight in the collaborations together with Chiara Branconi. So the reality is that still most patients see chemotherapy. Excuse me. And what clinicians like Chiara see is that there are some patients that respond remarkably, then we call exceptionals, long survivors. And then there are patients that rapidly progress and theoretically should not have chemotherapy at all, and those we call rapid progressors. And then, yes, this is the whole point, right? That chemotherapy still dominates the treatment landscape of cholangiocarcinoma. So chemotherapy really is the rate-limiting step that needs to be optimized. So we set up, or Chiara set up this group of patients that had rapid progressors. They have half the survival time, a median survival time, so less than 6 months. Whereas the long survivors, or the exceptionals, they have twice the overall survival compared to median, so 24 months. On the clinical workthrough of these patients, none of them are really remarkable. They are, not to be misunderstood, but they are really alike. There's no differences between them on the clinical, the blood counts, or on the systemic markers. Of course, I have highlighted two things that bongs out on the statistics, that is platelets and alkaline phosphatase markers, or marker for liver function, that are higher in the rapid progressors, but in multivariate analysis in all the following work, we do not see any association of these markers with the score that I want to discuss with you. As I said, the rapid progressors progress twice as fast. The exceptional survivors, long-time survivors, live twice as long as the median overall survival. So we got the diagnostic samples, and the pathologist indicated where the tumor core was. We also looked at the stromal content, the normal tissue in the biopsy, as well as other areas. But for the point of this part of the presentation, we investigated the transcriptome of the tumor cores. We used a technology called TempoSeq and defined the genes that separated these patients the best. There was 504 genes, and those we made a very simple score for, and we made the score of these genes across the patients to be able to go from this data to other data types, so we could validate this information in the current and historical data we have. Now, this is the only advanced and metastatic transcriptomic cohort that are on the way to be published, hopefully, in the near future, and we need more data. We need more samples of these patients to replicate it. So we had to go to resected patients to replicate this work, and there are currently five cohorts with clinical data available, our own that I presented in the past, so now we can actually still use 10-year-old historical data because we have a score, and then the different cohorts done job Yusukul and Nakamura that you have seen also yesterday. In all cases, we can significantly predict the rapid progressors, or the ones that are likely to be rapid progressors if they get chemotherapy, compared to people that would survive, or patients that will survive longer on chemotherapy. So that's all good. Now, I said that technology had developed over time, so we actually can answer some of the questions that we couldn't 10 years ago if we had done this study at the time when the ABCO2 trial came out, which would have been a fantastic place to investigate this in, actually. Because now we have single-cell technologies, because the tumor is not just cancer cells. There is a lot of other cells, immune cells that are part of the composition, and in cholangiocarcinoma, that is much worse than it is in its counterpart in the liver, hepatocellular carcinoma, for example. Now, the pathologists had indicated that this was tumor cause, so we would expect that the transcriptome signature, that the genes in this signature was mostly related to tumor cells, and that's what you can see. Most of the genes in the signature are related to the tumor cells based on signature deconvolution of single cell data. And that gets a little technical, but just trust me that it's using clean signatures from single cell data to indicate in this bulk tumor core what are the representation of these genes from these patients. So that's good. That's a control of it. The next thing is a lot of different immune cells, particularly myeloid cells. So there is a crosstalk that we would not have been able to understand 10 years ago between the tumor cells and the myeloid compartment. That might be the cause of the difference between a long-time survivor and a rapid progressor on chemotherapy before getting chemotherapy. So we did nanostring genomics DSP, which is a spatial approach, so indicating what are the cell types. So in this case, staining CD68 for the myeloid cells and pulling out this composition to the cell type to ask what is their gene build-up, if you will, and is there a difference between the rapid progressors and long-time survivors. So the RPLS signature predominantly originates, we know, from the tumor myeloid cell interactions, but the tumor-associated myeloid cells appear to be dysfunctional in the rapid progressors, and that can be one of the reasons, or that we believe is one of the reasons for why they do not respond as well to chemotherapy later on. Now, here we used the score to predict in single-cell data patients that would be considered a long-time survivor or a rapid progressor. And we did this because we now wanted to try to understand the communication between the different compartments of the tumor. So the bottom part is a lot of different analysis that was done in these single-cell data sets to try to understand this cartoon of communication between the cancer-associated fibroblast with the cancer cells, the myeloid compartment, and the T cells. And this is looked at from the view of a rapid progressive patient, not the long-time survivor. So anything that's in a long-time survivor has been filtered out in this analysis. We look at transcription factor activities, cytokine activities, and also ligand receptor interaction between these compartments. And as you can see, quite surprisingly, there's not much communication. Actually, there's no communication with the CAFs in this setting that are unique to the rapid progressors. So that's not where we have to go. And we already know from the signature that the myeloid compartment was our next bet of the role on why you are a rapid progressor compared to a long-time survivor. And there we see a high degree of communication going bidirectional, from the myeloid cells to the cancer cells and from the cancer cells to the myeloid cells. And we, of course, also see a lot of communications with the T cells, which is good for the approval of durvalumab. So the myeloid tumor cell interaction plays a key role in the rapid progressive response to chemotherapy. So actually, just to be a little selective and also to go one step backwards, one of the communications between the cancer cells and the myeloid cells, and from the myeloid cells to the cancer cells, are IL-6. So a few years ago, and that's the wrong citation, we published in a clinical cancer research paper on IL-6 as a biomarker for trying to understand or define its role in prognosis prior to therapy. And we looked in nearly 450 locally advanced and metastatic cases on the serum level of these patients. And IL-6 is a considerably better biomarker to predict death at 1 year compared to the current CA19-9. That was one part, so that's at a very fixed point. But if you look at the area under the curve on a continuous motion, looking at longitudinal data of these patients, you will see that the pre-treatment IL-6 is better predicting the short survival up to 1.5 years from diagnosis, whereas CA19-9 only starts to become predictive and a good marker for predicting survival after 1.5 years. And we know that a lot of patients, unfortunately, do not survive that long. That was a little bit of a detour, and there was basically a detour to indicate that work was then funded by AMMF as a continuation to try to understand the tumor phenotype and sensitivities that are triggered by IL-6 activation in cholangiocarcinoma. That's something that we are currently working on. The communication of why the cancer cells are provoked by chemotherapy, for example, to start expressing IL-6 that normally would come from the myeloid compartment otherwise. Now back to the rapid progressors. Until now, I've only been talking about intrahepatic cholangiocarcinoma, where we're able to predict in any data we have looked at so far if you are a rapid progressor or a long-time survivor in resected patients. So we looked at our own Pera-Hyler cases. So can we also predict in extrahepatic? And we cannot. We thought, okay, that is because it's a small cohort of 37 patients, so we collaborated with the Louvre group on their study on extrahepatic cholangiocarcinoma, Pera-Hyler and distal, much larger cohort, and we cannot predict. So it's not a cholangio-specific signature and score we made. It seems to be a liver-specific predictor. So this suggests that liver provides a unique microenvironment and engages distinct oncogenic programs to predicted by the RPLS signature that are significantly implicating patient benefit from chemotherapy. Now, if that is the case, another tumor type metastasizing to the liver, we should be able to predict. There's not a lot of data to go for, but there are two large basket trials, MET500 and POC570, that has targeted therapy and chemotherapy on metastasis. And we already know that the targeted therapy we're not predicting, we're predicting on chemotherapy because that's how the score was developed. It fails on the targeted, that is what we would have expected. But on their metastasis to the liver, any metastasis to the liver that receives chemotherapy, we are significantly able to predict both the negative correlations and the positive correlations of this score, and not metastasis to anywhere else in the body. So again, there is a unique environment driven by the myeloid cells in the liver that are able to predict the score of the predictability of this score. Now, a basket trial for each of the different indications have relatively few samples. So the next question raised was, so in a larger cohort where we have primary tumor type metastasizing to the liver, having transcriptomic data, having great clinical workup, so a lot of different requirements that are usually very, very difficult to get your hands on, can we now predict still the RPLS score? And for that, we contacted the ESCORT consortium here in UK. Their new EPOCH trial, and Bridgewater in the audience has been a co-author on that study. They were very gracious and gave us access to the clinical data, the transcriptomic data, so basically everything we needed on the primary colorectal cancer data and the colorectal cancer metastasizing to the liver. So if the score is to be what we think liver-specific and it's the liver environment that makes a difference, it should fail in the primary tumor type and be predictive in the liver mets. And that's exactly what we see. Fails miserably in the primary colorectal cancer data and predicts perfectly in the mets. And on a univariate analysis of this with different markers also used in the EPOCH trial, when we apply in our score to their data, it is much bigger, much more significant. It has a better predictability than all the other scores in that study. So to round up, and I will not go through all of this, but basically showing you the differences between a long-time survivor, what we believe is the difference between a long-time survivor and a rapid progressor, is really the microenvironment of the tumors, and particularly the myeloid composition, where we see a proficient anti-tumor immunity in the long-time survivor and an immunological inert microenvironment based on the myeloid cells in rapid progressors. And as Chiara highlighted in her ENSCA talk, and I've said a few times as well, this was a very, I think, fruitful collaboration and tight collaboration with her group in Glasgow, my group in Copenhagen, led by Com O'Rourke, who was also given the presentation much better than I did at the CCF. It was also a key collaboration with Max Solanti, pathologist from Italy, and many other people in the cholangio field, both here in UK and Italy, US, and Germany. And that's my team. And this is Com, looking very happy because he has submitted the paper, and we await the verdict. Thank you.